Well, we first started out approximately 4 years ago with our interest in looking at how environmental stressors affect, well, fish in general, but particularly rockfishes. Rockfish are extremely important commercial and recreational species along the entire Pacific coast. We were interested in monitoring the behavior and see where these animals are actually going real time and responding to the environment. For this project in particular, we have a physiologist, myself. I spent a lot of my time on the Antarctic Peninsula at Palmer Station, and that work really got me energized into looking at the effect of environmental stressors on animal function. We have a sort of fisheries ecologist with Steve.
I think I have that conservation bent. I've been lucky enough to work in estuaries. I love my salt marshes, kelp forests, a little bit coral reefs.
We have Mike, who is fantastic with the sensors and the fluid dynamics of a kelp forest.
I started out studying the physics behind how fluids interact and move when you go into the ocean. The physics just basically go crazy as well and become much more interesting.
We are using an acoustic array spanning our kelp forest here at Hopkins Marine Station. So we know that these fish are staying local. They have what's called a high site fidelity. So we capture them from the local kelp forest environment. We bring them into the lab. There are specific tags that we use, and we surgically implant these tags. We allow the animal to recover in the lab. We've had really good success with this. And then we release the animal. And then we're basically able to map in real time where these animals were and when they were there.
In the spring and summer, we get cold, deep water that comes up onto the shore. Due to climate change, those waters tend to be lower in oxygen and lower in pH, and that seems to be a trend that will continue.
So basically what the kelp forest array does is we can install sensors that measure water quality. Some of our most recent work has looked at the spatial variability of hypoxic low oxygen events, and then how species like rockfish respond to these events.
We're really interested in how does environmental variability affect an organism? How does it change what it does, where it lives, what it eats? Altogether, this is building a story about what does it mean when the environment changes to an individual. What does it mean for the ecosystem? It's all linked.
Knowing these energetics can also help us ultimately inform whether it's our fisheries and how we regulate that fishery.
I'm very interested in looking at ways to understand the mechanisms of nearshore processes to help inform ocean monitoring and inform other stakeholders who work in the nearshore.
We really believe this will be a breakthrough for maybe a lot of different researchers in going forward and how we can continue to monitor, some of these coastal species. I really find that, bringing sort of this team mentality to the plate, what we've been able to overlay with all of these levels of expertise and the actual data now is something that we could not have done individually.